Welcome back to lecture 11.2, where we'll be talking about interphase and meiosis 1. Before we get to that content though, let's talk about lecture question 11.1. Which of the following is true about the different life cycles, blah, 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 blah. If you said D, 1, 2, and 4, you'd be correct. Nicely done. Okay, cool. Moving on. Here we go. On to 11.2. All right, so we kind of covered this a little bit in the end of last lecture, but really briefly, with meiosis, we're going to expect to see it's going to be very similar to mitosis, except you do that PMAT process twice, and you're going to expect to have 4 unique daughter cells at the end that are haploid, so half the ploidy of the original cell, unlike mitosis where you have 2 daughter cells that are exactly the same as the parent cell. Okay, quick note before we get into the content here, specifically about the book. So the book has some really nice figures about this whole process. For example, figure 11.6 is a really excellent sequence of meiosis that you should probably take a look at. A couple of issues to point out though. Number 1, we actually don't talk about pro-metaphase. We just collapse that into prophase, okay? So ignore that extra step here. So aside from that, I like how this whole thing is nicely color coordinated between meiosis 1 and meiosis 2. You have, meiosis 1 is kind of orange or yellowy. Meiosis 2 is kind of a darker orange, maybe a salmon. And it's pretty nice and clean and simple, these kind of graphics for each of the stages. That's nice. Now, the wordage in the outcome category is a little bulky, and in other words, you could probably break that up into a few more categories or something, but overall it's pretty nice. However, I will not be using this. There we go. I will not be using this figure during lecture. Allow me to demonstrate why. Say I wanted a figure about anaphase. Now I could take that figure, crop out the rest of the image here, and then so it's only anaphase, and then blow it up so we have a nice clear image for the screen, right? So there you go. That's kind of what it would look like. Not bad, but it does have these stupid arrows left over, and that really annoys me. And I think all these other figures are going to have a similar, these stages are going to have a similar issue with these stupid gray arrows and kind of a obnoxious background. So I'm not doing that, okay? I have some slightly different figures that I'll be using for each stage, but don't sweat it. They're really similar, except of course the book talks about prometaphase, and we just jam that in with prophase 1. And I should say we jam prometaphase and prophase together, regardless of which prophase it is. Anyway, let's get into these steps, starting with interphase, which should be pretty familiar since it's exactly the same as in mitosis. We have the chromosomes duplicating during S phase and the centriole pairs replicating. The result, of course, is the chromosome composing of 2 sister chromatids, which are exact copies of each other, right? Now, at this point in humans, with a ploidy, normally have a ploidy of 2n equals 46, we have 92 chromatids, but you still have a ploidy of 2n equals 46, okay? Now, ploidy has not changed yet. Why? Again, same reason as with mitosis. Just because you have more of the same genetic information does not mean you have more information, right? Getting back to Harry Potter, we've only photocopied our books at this point. We've not added any new versions. So we've not changed the ploidy. Now, so this figure on our right here does a pretty good job at summarizing what happens in meiosis 1. As we can see, homologous chromosomes are separated. Now, in other words, the copies of mom's chromosomes have moved away from the copies of, use my fingers here, mom's chromosomes have moved away from the copies of dad's chromosome by the end of meiosis 1. This results in 2 haploid daughter cells, where you're going from 2n to 2 times n, right? Now, why are they haploid? We'll get into that. But think about the definition of ploidy and how many versions of each chromosome those daughter cells have. Okay, so from here on out, we're going to use this simple example you can see here. So there's, with these kind of chromosomes, the blue ones and red ones, representing different sources of those chromosomes. So, blue can be mom, red can be dad. I'll probably flip that back and forth about 8 million times, but I think you get the picture, right? Okay, so the first thing we want to do here to help kind of keep track of what's going on is count the chromosomes. So it's handy to do that so we can keep an eye on how these changes throughout the rest of this process. So first off, we have 1, 2, 3 different chromosomes from dad. Now, if we're only counting those, or only the kind of one set of chromosomes, that means our haploid would be, our ploidy would be haploid, and we write it as n equals 3. However, of course, these are paired up in homologous pairs with chromosomes from mom, right? So 1, 2, 3 more from mom here. Now, if we have 2 copies of each chromosome, one from mom and one from dad, that means what? 2 chromosomes, one from mom, one from dad. Right, we're diploid. So our math should be 2 copies of each of the 3 chromosomes, adding up to 6 chromosomes, or 2n equals 6. Also, the chromosomes have doubled at this point since they've gone through DNA replication, but they're still diploid, right? Since they still have the same number of versions of each chromosome, 2. So there's a long one from dad, long one from mom, medium from dad, medium from mom, short from dad, short from mom, right? Okay. Now that we have that all kind of sorted out, let's carry on with what is actually happening. Now, prophase 1 is the longest stage, and it's when chromosomes begin to condense, which is pretty much what we know from mitosis too. However, we have something a little different occur here. Now, when the homologous chromosomes pair up, like this kind of long blue one and long red one here, they align next to each other, gene by gene. This lining up process is called synapsis. Now, when this happens, structures called chiasmata can occur, where the genes from one set of homologous chromosomes actually switch or cross over with the genes from the other homologous chromosomes. So there's where the genes kind of switch or cross over. Maybe actually going back to our Harry Potter analogy can help a little bit. So here's our 7 Harry Potter books: the Sorcerer's Stone, Chamber of Secrets, Prisoner of Azkaban, Goblet of Fire, Order of the Phoenix, Half-Blood Prince, and the Deathly Hallows. We also have 2 versions of each book, the American and the British, making our total ploidy 2n equals 14. This would be in a single parent cell, right? Now, of course, this isn't taking into consideration that we've doubled our chromosomes through DNA replication, right? Of course, just because we've doubled it, our ploidy isn't affected. There's no new versions added yet, right? Then we form homologous pairs. In this case, the Sorcerer's Stone, known as the Philosopher's Stone over in the UK, would be considered homologous pairs, as they are both book 1 and have the kind of same basic storyline. Now, here we kind of have those 2 books, the British on the left and the American on the right. Now let's try expanding this out a little bit here. if we consider each chapter, we can kind of think of those in our analogy as like the different genes on the chromosome. And we can distinguish between the British chapters as red and the American chapters as orange. And also the American chapters will be italicized, so that's easy to keep track of too. Now, when the homologous pairs line up in prophase 1, our chapters can switch. So that means one of the sister chromatids of the British version has a few chapters from the American version, and one of our sister chromatids of the American version has a few chapters from the British version, like you can see here. Fun question, though. What would happen to the ploidy because of this? Nothing. Ploidy is the same, right? Any guesses as to why? Okay, right, yeah, it's because we still only ever have 2 versions of a gene. Only by adding, say, maybe a German translation of our Harry Potter book could we increase the ploidy. Now, if you're not a fan of Harry Potter, figure 11.3 is also pretty good. It's a nice representation of how crossing over occurs. It uses simple upper and lowercase letters to represent the genes from the different chromatids, and, well, it's fine, I guess, if you're into clean and simple examples. Obviously not as fun and exciting like the Harry Potter books, but sure, it works very nicely too, I suppose. Anyway, back to our actual chromosome models, which are now in metaphase 1. During metaphase 1, homologous pairs of chromosomes line randomly up along the metaphase plate, and microtubules attach to the opposite poles via those kinetochores. Sounds pretty familiar, right? Well, really quick and really important to point out, though, remember I said they randomly line up? Well, like you can see here in this diagram, that means some of the homologous chromosomes from the mom are oriented on one side, while some are oriented towards the other side. This is really important later on down the road. Now, actually, figure 11.4 explains this pretty well. In this example, we have cells that are 2n equals 4 that we're starting out with a kind of a long chromosome from mom and dad and a short chromosome from mom and dad. Now, how these randomly line up during meiosis, during metaphase 1 specifically, can result in very different daughter cells. For example, up top here, we have happen to produce daughter cells with chromosomes entirely from mom or entirely from dad. However, they were just as likely to line up in this scenario here, which produced daughter cells that have 1 chromosome from dad and 1 chromosome from mom, or, switched, right? Now, this is one of the ways you can start seeing diversity in offspring during sexual reproduction due to meiosis and how meiosis functions. Now, keep this process in mind when we get to meiosis 2. Okay. Now, after metaphase 1, we're following again that PMAT structure, prophase, metaphase, anaphase here, of course. In this case, we call it anaphase 1, where the homologous chromosomes are separated, which means that at the end of this, we actually have the start of our haploid cells, each of which likely has some kind of combination of chromosomes from mom and dad. Going back to my last point about the ones from mom and dad randomly aligning, this means at this point, you can actually have those different combinations of homologous chromosomes moving to either side. Now, if the homologous chromosomes separate normally, that's called disjunction. If they do not separate, that's called non-disjunction, and it would result in one of the daughter cells having an extra chromosome, while the other would have one less chromosome. Now remember, the sister chromatids remain attached to each other at this point. It's only the homologous chromosomes that separate at this stage of the game. Okay, getting back to Harry Potter. Using our British and English, or I should say British and American versions from before, the ones where we kind of flip the chapters, right? If our different copies of each version are our sister chromatids, right? And our two versions together are homologous chromosomes, then disjunction means the British and American versions would separate and move apart. Right? So when they move apart, normally this is considered disjunction. However, non-disjunction is when they go to move apart and one of the sets of homologous chromosomes doesn't separate. This would be called non-disjunction, right? And for some reason, one of our cells is getting two versions of Harry Potter Book 1, while another cell is getting no versions. Okay. Now, in telophase 1, we have a similar process to mitosis, where the nucleus reforms in each cell and the cytokinesis kind of splits the cytoplasm. But in meiosis 1, the chromosomes in each daughter cell each have two sister chromatids, which means that by the end of telophase 1, both cells are now haploid. Okay, let's maybe revisit our Harry Potter analogy and see how that works out. So if the blue line here is the new plasma membrane that's formed after telophase and cytokinesis, we can see that each side has two copies of our 7 unique books. But since there is only ever one version of each, we're considered haploid, and we'd write that as n equals 7 for each of these daughter cells. Now don't forget though, this is theoretically what it would look like, but more likely, because of how homologous pairs randomly line up on the metaphase plate, each cell likely has a mix of British versions and American versions, or a mix of sister chromatids coming from mom and sister chromatids coming from dad. But still, they only have one version of each book, making them haploid. Okay. This leaves us at interkinesis, which is a good point to kind of leave off here. But at this point, we have two daughter cells that are haploid, with still two sister chromatids for each chromosome. Make sense? Okay. Now, this is going to be a perfect place to kind of pause and then pick up again next lecture with interkinesis and meiosis 2. Okay. Which leaves us with one last thing to do. The lecture question. Just kidding. We're not doing that. What instead we're actually going to do 2 lecture questions at the end of lecture 11.3, because there's kind of some more stuff to talk about by that point. So stay tuned for that. All right, I'll see you in lecture 11.3. Bye.